as typical, they saved the best session for the last session to encourage everybody to stay. We've heard a lot of talk about autologous CAR T and allogeneic CAR T, but really this is the next best thing. And I'm really excited about today that I was able to chair this session. We have three excellent speakers, Ulf Grawunder, who's the CEO of T-CURX, Cecile Bauche, who's the CSO for Alaya.bio, and Bruce Levine, who a few of you may know, but is the Barbara and Edward Netter Professor in Cancer Gene Therapy at the Perelman School of Medicine at the University of Pennsylvania. So I'm really glad you're all here. It's going to be a great session, and hopefully we'll have some really good discussion, but if you'll just come on up.
Sure. Thank you. Thank you for the nice intro. Okay. Well, good morning, everybody, and indeed, well, great to have you here. I know that everyone is already excited getting back home and leaving the conference, but we hope that we can really give you a really nice insight about the next developments in the autologous CAR T field, which is, well, in vivo CAR T generation, where there's a lot of buzz around this. So I have a more educational presentation here for you today, where I would like to highlight the promises, but also the challenges with this modality. And without further ado, I will start with the first slide, which is actually a conflict of interest disclosure. I think this is a remnant from a previous presentation. So I'm board member on a couple of companies. I'm, of course, CEO mostly and board member and also a shareholder of T-CURX, a CAR T company from Germany, the wonderful federal state of Bavaria, where we also, I will give you one slide what we do at T-CURX because it relates to what I'm going to tell you about this morning. So I think in this audience, I don't need to highlight the enormous success of autologous CAR T therapies that have come along in the last couple of years. So we have now 7 FDA-approved CAR T products, which have fantastic response rates in patients. Just as one example here, the result of the CARTITUDE-1 study with Janssen's CARVYKTI, where you see almost 100% objective response rates and really 2/3 of the patients going into sustained complete responses. I mean, this is what keeps everyone really excited to have these fantastic efficacies and cures for cancer patients. Nevertheless, I think the dilemma is that there are very few patients that really benefit from these therapies. And you see here a table that the company Ori Biotech is constantly updating. So I checked it yesterday. And you see here that we are kind of approaching slowly the 10,000 patient mark per year, patients that are treated with these autologous therapies. I apologize, they also put the TIL therapy here on the bottom. And of course, the newest CAR T approval from Autolus is not yet on here because they will only start recruiting patients in 2025. But clearly, only a fraction of the P cells really benefit from these therapies. And therefore, the market, the global market is still relatively small with 3.7 billion. And even if you have fantastic growth rates projected for autologous CAR T or for CAR T therapies in the range of 40% per year. And if you look at kind of the projected global sales in 2029, $29 billion US dollars. If you calculate this still on the average cost per CAR T therapy of 400,000 US dollars, it's still less than 100,000 patients for more than 800,000 leukemia, lymphoma, and multiple myeloma patients globally out there. So even with this impressive growth, we are still capturing less than 10% of the global market. And I think something really has to change here because this cannot be the future. And part of the problem is, of course, the very complex centralized manufacturing process that is applied for all of the FDA-approved products. You see here on top, of course, the part that is done in the clinical setting, and on the bottom in green, the part that is done in centralized manufacturing facilities. I think I don't need to educate you about all of the complexities in the logistics, but also a huge bottleneck for the vector supply, because all of the FDA-approved products are manufactured with lentiviral and retroviral vector systems, and this is a very cumbersome process as well. It requires packaging cells that you cannot scale to eternity or to any scale. So that makes it expensive. And in some of the CAR T therapies, the vector supply seems to be the biggest bottleneck. This is certainly for some of the multiple myeloma products where there's not enough output on the viral vectors so that the products cannot be supplied to the patients in need. So the big elephant in the room is we need a radically new way to manufacture CAR T cells or to make CAR T cells available for patients. Again, you have the numbers here and the reasons why we have these limitations, the complications and limitations with lentiviral vector supply, the complexity in the manufacturing process. And that requires actually all a very high infrastructure investment in GMP cleanroom infrastructure, of course, highly trained personnel, also equipment that is expensive, and even the single-use material is not on the cheap side. And in fact, if I talk to clinicians, they always tell me, well, these $400,000 per patient, they all go to the pharma companies, but we do not earn anything in the clinic ourselves. We even, I mean, for us, it's a loss-making business. It's just a high prestige to offer CAR T therapies to patients. So I believe one needs to empower the clinicians to bring CAR T cells really to the patients in a point of care or clinical setting. And this is what T-CURX is also working on. As I said, if we have time, I present that one slide there at the end. And if you look at the patient numbers where CAR T cells now are developing towards to solid tumor indications and autoimmunity, then we are not only talking about 800,000 patients per year, but millions of patients per year. So how can this be manufactured with the current complex centralized manufacturing? I think it doesn't work. So it needs a radically new solution for CAR T manufacturing. And of course, very attractive there's the in vivo CAR T strategy. However, pharma companies are not embracing it immediately, so they have other strategies. They do massive infrastructure investment to beef up their centralized CAR T manufacturing capacities. But this is mostly, of course, in the high-income G7 countries. I think the rest of the world countries will have to wait for a long time until they get access to sufficient numbers of CAR T therapies themselves. Of course, biotechs, well, they are intelligent. They have nifty engineering solutions. So they are devices, things, automation, parallel processing. There are many companies working and making CAR T manufacturing, even in a centralized manufacturing setting, more efficient. Few names are here. But of course, also people are pivoting into allogeneic CAR Ts using other types of cells which are not alloreactive, NK cells, iNKT cells, gamma delta T cells, macrophages. People put CAR constructs into everything that is available as an immune cell in order to try allogeneic approaches, or of course the gene editing that you need with T cells as a starting material. Hello? Of course, in the academic setting, there has been a trend in recent years to actually get independent from the centralized manufacturing process and rather to pursue point-of-care decentralized manufacturing. I think many people know about the point-of-care manufacturing of CD19 CAR T products in Barcelona under an hospital exemption from the European Medicines Agency. But also this is slowly getting into the commercial space. Galapagos, in their most recent study, they have started the first point-of-care manufacturing study in Europe, and they've touted very much at the ASH meeting in December of last year about the advantages of having really decentralized manufacturing, point-of-care manufacturing, where the cell product is actually fresh in and fresh out and not frozen, and that they can supply the patient within 7 days. The CAR product, which does not require extensive bridging therapy. So clearly, these are all strategies. But I think one of the hottest areas is the in vivo CAR T generation, where you don't have any device, don't have any infrastructure. You essentially have a vector that you inject systemically into the patient, and all the magic happens in the patient. The CAR product is generated in the bloodstream, and there are essentially two different strategies out there. People are using either viral vectors, lentiviral or adeno-associated viruses, or most recently mRNA LNPs, because that is an established technology that we all know from the COVID vaccines, and that has been redirected for T-cell targeting in various ways. So very simply, I mean, if you look at it on paper, it's of course very enticing. There's only a short-term infusion into the patient. The patient doesn't even have to undergo lymphodepletion. Ideally, the vector finds the T cells specifically in the circulation. They expand, and there's not much that you need to do at the end of the day. And the CAR T cells find the cancer cells and kill them. So that's easy on paper, but of course it's not so easy to implement. It's actually interesting if you look, and I updated this chart yesterday, the amount of publications that have been published in recent years. You see there's a surge in the last 5 to 7 years. So it started with 2010. If you look in PubMed for in vivo CAR T, you find a single publication in 2010 and just a few dozen in the next 5 to 6 years. But then it really started taking off. And if I look now in January 22nd, that was yesterday, we had already 30 publications. We will probably get up to 50 or so, maybe 40, 50 every month. So we will exceed the 500 publications per year, probably in 2025. So this will go on. Extreme interest here in this area, as I said, mostly viral, non-viral vectors being used, liponanoparticles, polymeric nanoparticles, which encapsulate the DNA vectors, or viral vectors are also being redirected for in vivo CAR T generation. And as most of you know, I mean, this was already described scientifically in 2018 and also then 2019. Was the group of Christian Buchholz at the Paul-Ehrlich Institute in Germany, who published that you can use CD8-targeted lentiviral vectors in order to generate CAR T cells in mice that have been engrafted either with human peripheral blood mononuclear cells or with CD34 hematopoietic stem cells. And without going into details, they could show they can be generated in these mice, and they also target endogenous B cells. And they later showed in 2019 that when you engraft the cells with leukemic cells, then they can also be targeted by these in vivo generated CAR T cells. So in the mouse, that all works. And it took a couple of more years to show that at least in monkeys, that seems to work as well. This is one publication here from Umoja Therapeutics that are using a particular targeting and VivoVec particles, lentiviral vectors that target T cells. And when they do an intra-lymph node injection of these vectors in monkeys, they can see on the right side here that they get roughly, 49% CAR T cells. And you also see that they actually deplete the endogenous B cells in the monkey. So you have actually the ability to do this not only in mice, but also in monkeys. And of course, now the question is, well, does that work in humans as well? I mean, it's very promising, but as I said, there are also lots of challenges out there that are summarized here in this table. I mean, generally with the in vivo approaches, of course, you have to ensure that you get precise targeting of the T cells and not anything else. I mean, I think this is the biggest challenge here, the distribution, and that you're not targeting these vectors to essential organs or to cells where they can be harmful. So then the question is it also efficient enough? But I think there's also one significant challenge. Well, what happens if you target a CAR construct to a leukemic or lymphoma cell? Then you have the target and the receptor on the same cell. And of course, the CAR receptor has a signaling function. So you could produce kind of a super leukemia or lymphoma cell. So certainly you have to include certain safety switches to kind of be able to delete such super leukemia or lymphoma cells. I think that from the safety perspective, there are still quite some challenges here. Of course, lentiviral vectors have a natural tropism for hepatocytes. So a lot of the vectors actually end up in the liver. They might end up in other vascularized organs. So these are challenges. And then the challenges with mRNA LNP vectors is it's only a transient expression of the CAR. So you might need repeat dosing. That causes the toxicities of the lipo and nanoparticle components, especially the cationic lipids are known to be hepatotoxic. And repeat dosing, of course, both for lentiviral vectors if necessary and for mRNA LNPs causes immunogenicity. And if you work with AAV vectors, there might be pre-existing neutralizing antibodies, but any injection of a complex viral vector or mRNA LNP causes neutralizing antibodies and an immunogenicity reaction. So the issues with regard to safety, precise targeting, and also efficiency are still enormous, because the question is, can you target sufficient numbers of T cells endogenously, and is that army that you produce there sufficient to kill the billions of tumor cells in a leukemia or lymphoma situation, which you address if you expand the CAR T cells ex vivo. So that's the big question. Is it equally safe and equally efficacious in comparison to the ex vivo expanded CAR T cells? And can it work without lymphodepletion? Because clearly you cannot do lymphodepletion because you need the T cells in the body. So that is the question. But it's now moving into clinical validation. This is a table from late 2024, a publication by, I believe it was Mallard et al. I can't see it from here, where you see that companies like Umoja, Interius, etc. are moving into the clinic now. Either they said at the end of 2024, but I would say the year of proof of the pudding is this year. And in the coming years, you see there's also 2025 and 26 as target dates when these products move into clinical validation. And also you see here that massive amount of funding is flowing into these companies. So I cannot go through all of this, but in order to come to this Hall of Fame here, you have to have at least 100 million funding. Then you can get into this list of top 10 in vivo CAR T companies, and some of them have raised more than $500 million up to $1 billion of funding, either through venture capital funding or IPOs or secondary offerings. But clearly, the process goes, in my view, from centralized to point of care to bedside CAR T manufacturing and eventually to in vivo CAR T manufacturing, because you need less of the infrastructure requirements, and the manufacturing cost will be much lower. I'm getting the warning sign here already. I'm over time, so therefore I cannot present our technology here, but we are of course exploring more the bedside CAR T manufacturing, but we have the optionality with our transposon LNPs to also go into in vivo CAR T. And I come to the conclusion, that's the last slide. I'm pretty sure that the current centralized manufacturing is not a long-term solution for the supply problem that we have. It has to go into decentralized or eventually to in vivo in situ CAR T technologies that will transform the CAR T field for sure. I mean, there are huge investments already made, mainly in the United States. Unfortunately, in Europe, it's much more difficult to get adequate funding, but there is enough competitive pressure out there that something will result from this. And some big pharmas already engaged in that space. I mean, AbbVie has this collaboration with Umoja. Other companies are active here as well. So this comes to the mainstream. Of course, two main technologies, lentiviruses, mRNA, LNPs. We have to see which one wins. Still significant challenges, I mentioned that, and we will throw our hat into the ring here as well with our transposon LNPs. And I think with this, I thank you, but without looking into the crystal ball, I think it's very clear. In 10 years from now, CAR T therapies will look very different from today. And with this, I would like to close and thank you for your attention.
Thank you very much, Ulf. And thank you for actually addressing one of the first questions I'm going to ask is, what are the risks? For those of you who've come in late, so we won't have Mike Klinczynski from Charisma. Unfortunately, he fell ill after JP Morgan. And so with that, I'll introduce our second speaker, which will be Cecile Bauche from Alaya.bio.
So thank you, and I would love to thank the Ulf for his nice presentation, because like that I will not have to explain all the problems, solutions, and I could go directly to my technology, so sorry about that. So what we do at Alaya, so I will skip all the problems with ex vivo manufacturing, logistics, patients. And we really wanted to know how to do the best in vivo CAR T gene delivery system in the first indication to do CAR T in situ into the patient. Everybody understands it can go faster because you do not have all the manufacturing and everything, no ex vivo manipulations. It's supposed to be one systemic administration. We don't really know yet if we will have and if the other people will have to reinfuse or not. But let's say one systemic administration will be great. And the most important is that it's off-the-shelf drug. Cheaper, it seems obvious, as mentioned, no costly manufacturing facility, no cell expansion, no high QC. You still have to produce the vector or the LNP or whatever, but I will show you that it can be really cheaper compared to existing therapy. Shorter and simplified supply chain for sure. But we really wanted to find a way also to do what we call the better cells, because you are supposed to transduce cells in vivo and the patient will become his own incubator. And it's really important to really bet on the immune system of the patient to try to have the best response, which may causes difficulty to find the dose. To find the way to expand the cells into the patient, because all the patients, when you work with patient cells, you know they are all different and it's sometimes difficult to have, let's say, coherent results between donors or patients. But at least we try to find a way to preserve the phenotype of the cells of the patient, especially the naive and the memory phenotype. We think it's going to increase the persistence and the efficacy, and I will show how. And as mentioned, no lymphodepletion is needed, so it's always good for the patient. I will show you also that we can do a redosing, find a way to redose. So how we do that? We decided to work not with a full decorated lentiviral vector, not with a full LNP or polymeric nanoparticle, but trying to combine the best of the two technologies. The lentiviral vector inside is a fully normal lentiviral vector, which has everything to integrate, transduce, and express the transgene, but we produce it without the pseudotyping envelope. So the lentiviral vector is totally deficient for transduction. So you may say it's a lot of money to produce batches, to produce something which is not working, but we coat the lentiviral vector with polymers. So 2 layers of polymers. The polymers will have 3 functions. They will restore the ability to transduce the cells. They will protect the lenti in the bloodstream. So you will, and we will see that we also detarget from the liver, and you can covalently or click chemistry graft any kind of targeting agent at the surface of your nanoparticle. So we take the advantage of the best what can do the lenti and the best of the polymeric technology. For our first application, we have anti-CD3 targeting agent, and I will come on it. We have screened a lot of different targeting agents to find the best, depending on the application people want to do. So what we did, the first experiment we did is to compare back-to-back a regular GFP coding lenti that you have on the left. We put it on human quiescent T cells with no activation factors, no cytokine, nothing. And we wanted to see, as expected, you have a very low transduction by using lenti without activation. And we realized that with our nanoparticles, so same transgene, same quantity, same cell, same donors, just by adding a polymeric coating on the nanoparticle, you restore the ability of the lenti to transduce, but also you really increase the ability to transduce quiescent cells. So just by adding the nanoparticle on the quiescent cells, you already have between 20 to 50% of cells which are expressing the transgene. So you start at the beginning with the really highest quantity of fresh, or let's say with a good phenotype cells. We wanted to see if those cells, once they are transduced, they are able to kill the cancer cells. So we use the same kind of nanoparticle, but coding for a CAR. And we put it directly, so it's a non-targeting nanoparticle. We put them on human quiescent PBMCs. And you can see that you can reach up to 20% of T cells expressing the CAR. And when you put them in contact with cancer cells, you have a lysis. So once it's inside the cells, the lentiviral vector will behave exactly the same way that it is supposed to behave, and the same way then it is doing when you are transducing a T cell with a regular lentiviral vector. Once again, no activation factors, no cytokine. It's really the expression of the CAR that activates the cell once it meets the CD19 cells. Very quickly, we did a lot of experiments to see if we had a good expression, long-term CD19 CAR expression. But if we move to in vivo efficacy study, we injected some cancer cell IV into immunocompetent animal. For us, it was really important to work with immunocompetent animals. And so it's a mice CD19 CAR in this kind of model. We injected the cancer cells, check for the presence of cancer cells into the blood, and injected either the vehicle or the nanoparticle. And you can see that once you inject the nanoparticles, the mice are really well surviving. We decided to make a challenge with the cancer cells to see what was happening, and also another challenge with the nanoparticle to see if something was happening in terms of toxicity, and we saw nothing. So no lymphodepletion was required. Some clinicians want us to make a small lymphodepletion, maybe to help the growth of newly generated CAR-T. It's possible, but we didn't evaluate it here. No pre-activation of the PBMC before the injection of the nanoparticle to increase the transduction. It's possible to do it. We will evaluate what is the best for the patients and maybe to reduce the dose. But here in this experiment, it was not done, and no cytokine was added during the course of the experiment. And we evaluated also the toxicity. We saw no problem of toxicity of the animals, no weight loss or whatever, but also no liver enzyme increase, no visible hepatotoxicity, and no cytokine inflammatory cytokine were generated either around the injection and during the course of the experiment. Some group of mice have been receiving up to 15 injections of the highest dose of the nanoparticle without showing any problems of toxicity, which is not the case all the time with LNP, for instance, but polymeric nanoparticles are really great. Here you have the following of the cancer cell into the animals I've just shown you. So on the right you have the control, and on the left you have the one animal in the treated group. Here you can see that once you inject the nanoparticle coding for a CAR in an immunocompetent animal, so not an efficacy model, you see that you have the increase at day 10, exactly the same increase proliferation that you have when you are infusing CAR-T generated ex vivo. So it seems that the proliferation just occurs the same, apart that you do not have to make any lymphodepletions. What we realized, and it was a point previously described, that where are the nanoparticles going? And it was really important for us to try to understand where they were going. So we infused a lot of regular lentiviral vector in immunocompetent animals and compare it to the nanoparticle non-targeting, really to see, for us it was the worst case scenario for biodistribution. And you can see that the regular lenti, as expected, is going directly to the liver, so you find integrated sequence into the liver. You also find some sequences integrated in vascularized organs, so you can find them in the spleen, bone marrow, sometimes in the lungs, which is not the case at all with our nanoparticle. The polymer is really detargeting from the liver. We found no integrated sequence in the liver, and everything in blood cells only. So we had done all the work with a non-targeting prototype to really work on the safety and the kind of cells we transduce. And now we move to the targeting one, nanoparticle. So we have two different kinds of targeting agents depending on what we want to do. Some indication will require a totally non-activated targeting agent, and I'm thinking about autoimmunity or things you don't really want to add activation on the patient. And we have been screening internally for an aptamer, an anti-CD3 aptamer, who is a really small pieces of DNA, no activation of the T cells, no immune reaction against such a small fragment of DNA, and it also has a very good KD. So it will be grafted at the surface of our nanoparticle, but for other indication, and some people really want to have either a big activation or a smaller activation, we've been screening for anti-CD3, SCAV or VHH, all humanized and human, because we didn't want to start with I should not say old, well-known targeting agents, which are not humanized or human. And we started with 21 protein-based anti-CD3. And at the end, we only found 9 which are fitting our expectations. It's interesting because on the right, you see they all have the same profile. So they are coming from commercial academics, a lot of different providers, and they all give the same profile. So very good at targeting CD3 positive cells, including NK T cells, no monocytes or B cells, and no other binding on other kind of cells. And we have the choice in the KD. So it goes from 0.6 to 200 nanomolar in KD. And it's really interesting because we do not know yet how it will behave on a 3D object. So we will have to work on the avidity. We don't know yet if it's good to have a high affinity or a lower affinity. We don't know. So this is why we have a range of evaluation depending on what we want to do and how fast we want to transduce the cells. We have a collaboration with Dr. Michel Sadlian because he will do all the translational work. So we did the tech transfer when Michel used to be at MSK, and now he's working at Columbia. So we have been transferring everything, and now we work with Michel at Columbia. So he will do all the translational work, mice evaluation of the phenotype, everything, comparison back to back with the CD19 ex vivo generated CAR-T. But we also licensed the 1XX sequence, so which is the sequence in the intracellular domain, which is allowing to lower the dose at 10x with the same effect than regular CD19 CAR. And I think that for in vivo application, it's really important to start with something which is already working, let's say, 10 times better than the ex vivo generated CAR. We have been working a lot on the manufacturing because it's good to have a good technology, but if you fall back in the same problems of manufacturing that used to be, it's not really efficient. So it's a microfluidic based manufacturing process. So you have the CAR here. We produce a lentille. We produce 2 kinds of polymer, the polymer with and the polymer without the targeting agent, knowing that we will play on the density between the non-targeted and targeting polymer. And we are using exactly the same kind of equipment that have been used for the COVID vaccine to produce the nanoparticle. So I should not say it's easy, but you just have to plug your polymer and your... The only point is that it has to be done in aseptic conditions, because the lentille, once it's coated, you cannot filter it anymore. So I should say it's the only point that can be, let's say, more difficult to handle. We've been interacting with regulatory agencies to present this way of producing and to present the data and to present the product. So we presented it to EMA, but also MHRA, and we plan to present that to the FDA this year, based on the data that Michel Sadlian will generate in his lab. Let's talk about cost of goods. So I'm sure there is a lot of mistakes in this slide, because we try to evaluate how much it costs to make a lentille with a polymer and a targeting agent. So we started with the real prices of GMP batches of lentille, polymer, etc., and tried to see how much it was costing. And we did it also top down and try to understand with the price of a cure and try to go down. And I have to say we arrive in the same range. Which is less than 10K for a dose. The dose is based without the 1XX, so maybe we have a bit flexibility, but what is interesting is that it only relies on the cost of the lentille. On the cost of the lentiviral vector, which is representing the highest cost. And sometimes we are asked, but what about the comparison with RNA? Maybe RNA are cheaper, whatever, but when you talk about long size RNA to do either transfection or LNP, we try to compare back to back, and we are cheaper than when you want to use huge RNA to make transfection or to put them in LNP when you work with complex CARs, especially the one that you have now for, let's say, having a CAR and a CCR or a CAR armored or whatever. So we are still cheaper than long RNA fragment. What is interesting with this technology is that we can use it in vivo, but we also can use it for extracorporeal application. So with all the machines that you have been seeing right now, you can pick up the cell of the patient, do the transduction and reinfuse right now. And with this technology, in a few minutes, you have a lot of cells which are already transduced and that will express. So it can be also a solution to in between the regular ex vivo manufacturing and the in vivo manufacturing. It can be interesting to have this kind of technology, either to move faster with a candidate existing that have been manufactured ex vivo, or to make a smooth transition between the ex vivo and the in vivo application. And as mentioned, it's a flexible platform. It's easy to change the transgene compared with decorated lenti because the lenti is not changing. It's even cheaper to produce lenti without the VSVG protein because your yields of production are higher. So we were surprised to have a 10x fold of increase in the bioproduction. The coating is the same because the lenti is not changing its surface protein. So coating whatever the transgene is inside is not a problem. And the click chemistry allows you to graft any kind of targeting agent at the surface. And what is interesting is that the click and the bioconjugation of the targeting agent with the polymer is done before the coating. It's not done like a LNP after the coating. So you really can manage the density. You know exactly what you put at the surface of your nanoparticle compared with grafting something after or making a decorated lenti. Here we are. So thank you.
Okay. So our last speaker, then we'll have a Q&A, will be Bruce Levine from University of Pennsylvania. Thanks, Bruce.
Thanks, Tony, and thanks to the organizers for the opportunity to present. I have a financial interest due to intellectual property and patents in the field of cell and gene therapy, and that conflict is managed in accordance with University of Pennsylvania policy and oversight. So in vivo CAR T, or some equivalent of medical intervention, was envisioned in 1966. And the film Fantastic Voyage, based on the book. So where are we going? And Ulf and Cecile both provided excellent introductions and talks, but let's think of the challenge of scale. We have approvals in leukemia, lymphoma, and myeloma. You have approval in solid tumors, you got to multiply the patient numbers by 10. Same thing in autoimmune disease. You have to multiply by 10 compared to blood cancers. And what happens when we go into fibrosis diseases, gene-modified stem cells, hemoglobinopathies, and rare diseases? So I would posit there are not enough humans on Earth to manufacture ex vivo those cells. Now, there is very fine work being done on allogeneic approaches and induced pluripotent stem cells, but could we leapfrog those approaches with an off-the-shelf autologous approach? Now, the in vivo landscape, as Ulf showed, is evolving rapidly. This is just from a report that I received from the Beacon Group, tracking the in vivo CAR therapies disclosed per year on the left, and then by delivery system on the right, and you see the breakdown viral, non-viral, and within non-viral, there are different types, and I'm going to talk to you about TLMP, which stands for targeted lipid nanoparticles. There are, and maybe this slide is also outdated with things changing so quickly, as of, I think this was 2 months ago, clinically active in vivo CAR T. There was a company I hadn't heard of, T-CURX. Many of you know of Myeloid, Interius, and Umoja already in the clinic. So where did things start as far as what I'm going to share with you? This came from an elevator conversation. We developed CAR T cells at Penn. We developed the technology behind the COVID vaccine. So what if you marry the technology of CAR T cells and the COVID vaccine, and Joel Rurik and Haig Akajanian and John Epstein's lab developed this in collaboration with others. This is a lipid nanoparticle encapsulating messenger RNA, encoding a CAR. That lipid nanoparticle is targeted by conjugating an antibody to the surface. In this paper at CD5, I'll also show you data with CD8. Now, the model system here was an induced model of cardiac fibrosis. So the CAR targets fibroblast activation protein. And now you can understand why having mRNA as temporary is useful, because you don't want to permanently target fat, because then you have an impact on wound healing and other tissues. So here's the model of cardiac fibrosis. It's infusion of angiotensin II and phenylephrine. And here, this is a CD5 targeted LNP FAP CAR. Now, on the left, you see measures of cardiac function. The gray is the saline control, and then the tan is the induction of cardiac fibrosis with saline, and then the red is with the targeted FAP CAR, and you can see the return to normal of these indicators of cardiac function. And the histology is shown here on the right, and you can go across and read the key there. So I'm a co-founder of Capstan. That's one of my disclosures. This is an evolution from the COVID vaccine technology in terms of the LNP formulation to reduce delivery to the liver, improve biodegradability, and maximize RNA delivery. And this can have applications not only in fibrosis, but also in autoimmunity. Again, having a temporary CAR is beneficial. You just want to reset the immune system, potentially settings in oncology, in measurable residual disease, and with the integration of gene editing into blood gene disorders. And so the précis here is that you could give multiple infusions just for how much you need. Again, autoimmunity, you want temporary. Fibrosis, you want temporary. Even in B-cell malignancies, if you're able to induce a remission with RNA and then remove that or it degrades, then you have the return of B cells and the absence of long-term B-cell aplasia and necessity for gamma globulin supplementation lifelong. Now here I'm showing you the evolution of the LNP. The tool is the COVID LNP, the Capstan LNP, and it's over here. Okay. So now we're looking, this is a naked COVID LNP. It targets the liver. There's also going to the spleen. And this is the COVID vaccine targeted. But now look at the Capstan designed LNP detargeting the liver, and because it's targeted, it's going to the spleen. So another way to visualize this is shown here. And on the left panel, this is measuring in the liver CD45 negative cells with a reporter gene mCherry. And so lower left is better, and you can see in green is the CD5 targeted Capstan LNP. In yellow is the untargeted COVID LNP. And then on the right-hand side is the opposite. This is in the spleen. And now upper right is better, and you can see the Capstan LNP in the upper right and the COVID formulation along the bottom there. Okay, so let's start in vivo models and humanized mice. And this is targeting CD8 in this case. The orange on the left is mCherry, and then the anti-CD19 CAR. You can see the effect within 3 hours. You see virtually complete B-cell aplasia. And on the right, that shows you the correlation with the CAR expression in CD8 T cells. So now let's go to monkeys. And this is a NHP experiment. This is a dose escalation from 0.3 mg per kg all the way up to 1.5, administering 3 doses. And you can see this dose-dependent and escalating increase in CAR-positive cells. Now, on the left is CD8. On the right is CD4, so that's your control. Some activated CD4 cells do express CD8, so that's why we see a little bit after the 3rd injection. Now, this is really key, and particularly in autoimmunity with the bispecific antibodies, you don't get that deep tissue depletion. This is showing you B cell count up top at the escalating doses with 3 doses. But in the spleen, and you can see the reduction, this is immunohistochemistry staining of CD20, and so you can see the absence of CD20 expressing cells in the spleen after this regimen. And the prior slide was with 3 doses. This is with 2 doses, 1.5, or 1, and then 2 mg per kg. Again, within hours, you can see the induction of B-cell aplasia up top, and within the secondary lymphoid organ, spleen, bone marrow, and lymph node on the bottom. So let's talk about the autoimmunity application. What we want to do is wipe away the pathogenic B cells and have a reset and return. And the way that we're measuring that is by looking at the different stages of B cell maturation, all the way from naive up to switched, class switched. And so as you go from, let's say, light green to purple, you're going from naive to mature. So then when you, the baseline is shown on the left, and then what you want to see is more of the green than the purple, and in fact, we do see that. So to summarize in this rapid, brief presentation, what I've been able to show you, I hope, is that we've been able to achieve reprogramming of specific cells depending on the targeting CD5 or CD8. We have rapid T-cell engineering and B-cell killing within hours in blood and secondary lymphoid organs in mouse and dozens of NHPs. Application in certain oncology indications and designed to facilitate access for a large number of patients. And so this will be tested in humans this year, and we're looking at expanding to those other indications. I just also want to show, again, another indication of how rapidly things are evolving is the number of abstracts at ASH. So in December, just list for you some of the in vivo abstracts that were presented. And for something that's a little bit different, what was hot that you were not expecting? So this is a paper that came from Marco Ruella and Mayan Levy at Penn, showing that a ketogenic diet enhances CAR T cell function. Now, how did this come about? Well, the question was, okay, in cancer, does your diet affect your immune cell function? And so Mayan Levy did this in a colorectal model in mouse. And okay, Western diet, high fiber, ketogenic, there was one other, and ketogenic 1, right? Okay, so there's the link to the paper. And then they isolated the particular compound, which was beta-hydroxybutyrate, and then you can supplement mice with beta-hydroxybutyrate, showed that that increases immune function and tumor control. So it's really cool study. And how does this apply to in vivo? Well, it could apply to ex vivo and in vivo. So what if you give patients beta-hydroxybutyrate and then you give them the in vivo CAR? You're improving the function of the T cells. and then that aids in the in vivo CAR. One other thing I want to point out, so because we've treated thousands of about 900 clinical trial CAR T patients and maybe 1,000 commercial CAR T patients, so we could do retrospective analysis of patients who responded and show that the serum beta-hydroxybutyrate concentration positively correlated with CAR-19 expansion. Okay, so there's biochemistry involved that I'm not well versed in because I had to take biochemistry twice. So I'll point you to the paper and acknowledge that I'm a representative of a large team of people at Penn at the Center for Cellular Immunotherapies, directed by Carl June, funders, initially philanthropy, federal, and industry, thanking our study participants, and the in vivo T-cell engineering, the Capstan founders on the left and leadership on the right. So with that, I'll stop and thank you for your attention.
So, Ulf, I want to thank you very much for setting up the stage, because that was going to be my first question about the challenges, and you had outlined several, particularly with regards to safety. And so I want to turn to both Bruce, you and Cecile, to tell us a little bit about how your technologies are addressing some of the safety challenges that Ulf had recommended. And I guess I'll start with Bruce, since you're right next to me.
Yeah, so one part of the safety is the mRNA that degrades and the CAR goes away. So if we're looking at targets like FAP, that's going to go away. If we're interrogating a new target, let's say a solid tumor target, then we can use RNA, and we have, going back a dozen years, we did mRNA electroporation ex vivo to investigate mesothelin as a target. So one part of is the RNA. Another part I mentioned on one of the slides, which is enhancing the biodegradability of the LNP. And of course, as Ulf and Cecile mentioned, we're looking for potential immunogenicity. We haven't seen significant. I mean, Capstan and our Penn collaborators haven't seen that. So
Have you done repeat dosing, though?
Yes. So I showed you with 3 repeat doses and a slide with 2 repeat doses. I am not sure of the maximum number of repeats that have been done, but that's been part of the farm tox studies.
So,
yeah, maybe I can comment. And it looks like you have all figured it out, right? Because I think the main challenges for the in vivo therapies that we've highlighted is the tropism for hepatocytes, or also the distribution to highly vascularized organs, which you have actually shown in your system. You seem to have under control. The general toxicity of the, let's say, if you have lipid nanoparticle components that usually prevent repeat dosing at more than 3, 4, 5 times. Usually also what we have seen in studies from companies working with mRNA LNPs is that the efficacy goes down the more often you repeat. So you get less cells because there are autoantibodies that are being produced. Therefore, I'm grateful that you did experiments in fully immunocompetent mice, showing that you're apparently not having these immunogenicity issues. So congratulations on that. But I've highlighted and I picked out one of the aspects that I find for the in vivo applications most dangerous. And I mentioned this, which is what happens if your CAR construct hits the tumor cell with exactly the target on the same cell. So you have basically, it's not even autocrine or paracrine, it's you have the signaling molecule, the CAR receptor with strong signaling moieties on the same, on the cell surface of the leukemia cell that has the target. So you need to make sure that this is not happening in patients, because then the problem of secondary leukemias is much higher than what you have traditionally due to insertional effects of lentiviral vectors, where the FDA has issued a black box warning. But then you have a higher frequency or a threat of secondary leukemia. So you have to install certainly some safety measures or kill switches into in vivo vectors to make sure if this happens, you have a way to interfere there. Maybe I stop here, and maybe you have solutions for everything. It looks like
I would love to.
So it's
No, but we are lucky enough to have more than one tool to try to understand how to handle the toxicity, but you never know. So this is why we have to use immunocompetent animals, humanized animals, NHPs, and you try to do your best to make a lot of generation of data, but you never know what will happen in human with such technologies. So this is why we also try to, and I think everybody tries to really reduce the dose to be sure that nothing will happen. And the problem we thought we had 5 years ago or 3 years ago are not the same right now. For us, when we started, we focused, we didn't want to have that when you produce lenti, you have CD19 CAR at the surface. And this is, for us, was something we didn't want to have that. We really wanted to say, well, we will inject a lenti with a CD19 CAR and you will fall in all the problems you don't want to have. So this is why we started thinking about coating it and removing the CD19. And now the problems we have are different. And what the people are doing now in vivo will teach us how can we handle what will happen. And this is why I'm really happy people injected, product into human, because we will know what happened and what we can do to do not better but differently, or to handle the problem. So it's a work in progress since 10 years now for everyone.
May I add something?
Yeah.
So maybe one solution is that you mentioned this, Bruce, that currently we are trying to leapfrog the complexities of the centralized manufacturing going directly in vivo. And you mentioned it also briefly in your presentation, and I highlighted it, but at the end I didn't have enough time. I think it's absolutely worthwhile thinking about the extracorporeal CAR-T manufacturing, because that takes away the risk of the toxicities of the vectors. So you have a controlled kind of process where you take the cells out of the circulation of the patient, but at the bedside of the patient. So essentially really in a system where it's almost like in vivo, but it's extracorporeal. And then you can wash away any kind of toxic components. You have a little bit of a quality control there. You're not risking that you're blindly injecting the vectors and not knowing exactly where the vector targets and what it hits. So I think maybe one should do a little less leapfrogging and approach this kind of in smaller steps. And this is the strategy that we are actually preferring in our company.
So you're advocating a point of care system which is using an extracorporeal device.
Yes.
For doing the...
Yeah, essentially like an apheresis machine or dialysis machine that you roll to the bedside of the patient. You do your thing within 24 hours. You have control of the process, right. But it doesn't require a complex GMP.
So this reduces this risk about the leukemia.
Exactly.
Bruce, I was...
let me just add something on what Ulf raises, which I think is worth considering, especially for a gene editing approach, where you really worry not only about off-target within the genome in a particular cell, but off-target to cells that you do not want to gene edit. And then you have a much higher, LNP or virus to target cell ratio. Now, I did see, was it this morning or yesterday, that there was a Chinese company that did the first in vivo gene editing. So I'm not sure I would sign up for that, but I wouldn't jump out of a plane either.
Yeah.
So,
yeah. Well, we have to see which of the strategies is really making bigger progress. I mean, currently, the biggest funding and most funding goes into the in vivo approaches. There's no question.
Right. And again, we still don't know about the safety risks. We probably won't get a better understanding until the next few years. And the numbers of patients, how many patients are in these studies? Bruce, you had cited some clinical studies.
Oh, you mean the in vivo studies?
Yeah.
Well, they're starting small. I have to look them up. My estimation is in the 10, 20, 30 range. But like all first-in-human studies, there are cohorts and there are stopping rules and dose-limiting toxicity rules. So I think based on the press releases, we'll probably hear about every patient after every patient is dosed, but we'll see.
I wanted to go to this concept of the personalized medicine, and where do you guys think this is going to be in 10 years? I mean, Ulf, you clearly think this is going to take over the field. Cecile, what are your thoughts in terms of the different indications where...
When you see all the patients that we need CAR-T or therapy, as mentioned, you have autoimmunity, you have cancer, you have gene therapy, it will be impossible to just produce, as mentioned. So if you want to treat this patient, even if you don't think about doing it at the bedside or whatever, you will never be able to produce all the vectors and all the T cells and everything. So you need to find solution. And for me, in vivo is a really interesting solution as far as it's done with the safety concern and everything. But it's going to change everything. And as you mentioned, within the next 10 years, we will see things totally differently. Yeah. By the way of treating patients at their bedside at a cheaper cost, I hope.
One could even say, I mean, currently the way how the CAR-T therapies or other cell therapies are produced is a total disaster for everyone. It's a disaster for the pharma companies because they hardly recoup their investment and therefore have to charge very high prices for their therapies. They're not getting enough therapies to the patients, so it's frustrating for the patients, it's frustrating for the doctors, and the healthcare insurance system has to carry all of that high expenses. So nobody's winning, everyone is losing out, and there's a huge disconnect between the demand and the supply. So in 10 years, it will look totally different because everyone is aware of the situation. There are 20 different solutions to that. I think I put all of this in the slides, but couldn't speak to every solution. In vivo is one, but I mean, there are other solutions out there as well. And it will, in 10 years, it will look totally different. It will be economical. It will be broadly applicable and accessible to patients. That has to be the objective.
Yeah, so there's, if you think of it, a difference between a dream and a goal, right? A dream, you can dream forever. A goal is something you strive to attain. And I remember, I don't know if you remember where you were, reading the Intellia paper in Science on in vivo editing of transthyretin amyloidosis. And I almost fell out of my chair because it happened, it was specific, and there was a dose response. So then you begin to think, if it can be done there, then it has to be repeated with potentially different platforms.
Well, thank you. Is there any questions before we run out of time?
Hi, nice talks. It was really good. You mentioned a lot of solutions, and a lot of solutions will exist. Seems like there is a coexistence of technologies for some time. Is it really 10 years that one technology will switch to the other, or is it like you have an a la carte of different kind of options and then you pick based on the indication and the cost, because cost, hopefully with automations and all, will come to some reasonable place. So what are your thoughts on coexistence, and what does the time frame look like? Yeah. Is it a complete shift in 10 years or coexistence?
No, it could go faster, actually, with all the funding that is involved there. I think we will see transformative developments in the midterm or short term. I just said 10 years because then I'm on the safe side. I think in 10 years for sure it will look different. I don't know what that's in 5 years. But also I would like to mention, going back to the kind of manufacturing dilemma we are currently in cell therapy. I've been around for a little while and have started various companies. My first one was in antibodies, therapeutic antibodies. 25 years back, there was a huge challenge producing antibodies cost efficiently. Now it's a total commodity. So, I mean, in 25 years, the world has totally changed. It's now, as I said, commodity, a 250 billion market of therapeutic antibodies. This is where cell therapy will come as well. And it will include, well, the better CMC manufacturing solutions and potentially in vivo. I think that's very clear to me. But whether it's 5 or 10 years, I can't predict. 10 years for sure.
You may be able to choose depending on what you need. You may not need a lentiviral vector or integration vector for what you want to do, and LNP or RNA is better. So I think you will have some specialization depending on what the patient really needs. The problem that sometimes now you don't know what he needs and what is the best to cure the patient. But in a few years, we will know better, and you will be able to adapt. So it's a kind of personalization, but a high level compared to what we call personalized now.
Do you have any additional questions? Well, thank you very much. Okay.
Yeah, one more.
One more. Go ahead.
Ready to go home. I was just wondering if you could comment on the ethical dilemma, if you may, because I feel like the whole in vivo thing feels a little scary to me versus, taking it out and doing what we have to do and doing all the testing that we feel like we need to do to make sure it's safe, even if it were to cost $100,000 versus $10,000, I struggle to kind of come to grips on what that may mean from a safety perspective.
Well, the data may drive your decision. And if all things being equal and you feel safer with an ex vivo, will your insurance company pay 10 times the price? That's where we may be in 10 years.
But I think you have a valid point.
It's just one patient that dies from Vivo, where they otherwise could see.
I think you have a very valid point. Therefore, I mentioned before that maybe doing the leapfrogging from centralized, extremely well-controlled and quality-controlled manufacturing process, which is cumbersome, expensive, and really not a model for the future, to directly going in vivo with all the safety concerns. I always apply for me, the litmus test is always, would I enroll in an in vivo CAR T study now? I would say no. I think, a lot of work has to be done, and I think your concern is absolutely correct, and therefore I advocate a little bit to rather move into the point of care, maybe bedside CAR T manufacturing area as well, where you have at least a little bit more control about the process in comparison to just injecting and hoping for the best.
Yeah, and I think it's not going to be a switch
Either
over
or, exactly.
at the 10-year. There's going to be a lot of overlap and choices for patients.
Exactly. And that's the good news. And maybe that's a good concluding
I mean, that's
word.
the good.
There are choices for
There's gonna have
patients.
more choices for patients. Thank you very much.